Common questions on the immune system, A-level, AQA. These are taken from AQA Biology past papers from their website. Explain how HIV affects the production of antibodies when AIDS develops in a person. Less or no antibodies are produced because HIV destroys helper T cells or reduces the helper T cell numbers, so few or no B cells are activated, or few or no B cells undergo mitosis or differentiate or form plasma cells. 2. Bacterial meningitis is a potentially fatal disease affecting the membranes around the brain. Neisseria meningitis is a leading cause of bacterial meningitis. In the UK, children are vaccinated against this disease. Describe how vaccination can lead to protection against bacterial meningitis. Antigen on surface of the bacterium binds to surface protein or surface receptor of a specific B cell. B cell divides by mitosis or produces clones. Stimulated by cytokines by T cells, B cells or plasma B cells release antibodies. Some B cells become memory cells. Memory cells produce plasma or antibodies faster. 3. When a vaccine is given to a person, it leads to the production of antibodies against a disease-causing organism. Describe how. The vaccine contains antigens from the pathogen. A macrophage presents the antigen on its surface. A T cell with a complementary receptor protein binds to the antigen. The T cell stimulates B cells with complementary antibodies on its surface. The B cell secretes large amounts of antibody. The B cell divides to form clones, all secreting the same antibody. Question 4. Describe the difference between active and passive immunity. Active involves memory cells, whereas passive does not. Active involves the production of antibodies by plasma cells or memory cells. Passive involves antibody introduced into the body from outside or a named source. Active long-term because antibody produced in response to the antigen. The passive is short-term because the antibody is broken down. Active takes time to develop or work, but passive is fast-acting. Question 5. Vaccines protect people against disease. Explain how. Vaccines contain antigens or dead or weakened pathogens, or antigens that are dead or weakened. 2. Memory cells are made. 3. On second exposure, memory cells produce antibodies or become active or recognise the pathogens. 4. Rapidly produce antibodies or produce more antibodies. 5. Antibodies destroy pathogens. Question 6. Some white blood cells are phagocytic. Describe how these phagocytic white blood cells destroy bacteria. Phagocyte is attracted to the bacteria by chemicals, or they recognise the antigens and the bacteria as foreign. They engulf or ingest the bacteria. Bacteria is enclosed in a vacuole or phagosome or vesicle. Lysosomes fuse with or empties enzymes into the vacuole. Bacteria is digested or hydrolyzed. Give 2 cell types that trigger an immune response. Cells from other organisms or transplants, abnormal or cancerous or tumour cells, cells infected by a virus, antigens on pathogens. Question 8. Some people eat red meat for many years without having any reaction, then have an allergic reaction to the alpha-gal in red meat. An allergic reaction is caused by immune response. Draw a label diagram of an antibody and identify the specific alpha-gal binding site. You need to draw a Y shape showing 2 long and 2 short polypeptide chains in the correct position. So your 2 long polypeptide chains should be the 2 inner ones, the 2 short ones should be on the outside. Alpha-gal binding site should be labelled at the ends of the branches of the Y of the antibody. The variable region should be labelled, which is at the top of the Y, each arm of the Y. Or a constant region labelled, which is the bottom of the Y. Or you can label the disulfide bridge or bond. What is a monoclonal antibody? It is antibodies with the same tertiary structure or an antibody produced from identical or cloned plasma B cells or B lymphocytes. The use of monoclonal antibodies in medicine. Targets or binds or carries drugs or medicine to a specific cell, antigen or receptor, or blocks the antigen or receptor on cells, except cancer or disease cells as a specific cell. Describe the role of antibodies in producing a positive result in an ELISA test. The first antibody binds or attaches that is complementary to the antigen. The secondary antibody with an enzyme attached is added. The secondary antibody attaches to the antigen. I would usually add also that it is washed in between time to remove any unbound antibodies. The substrate or solution is added and the colour changes. 12. Describe how presentation of a virus antigen leads to the secretion of an antibody against this virus antigen. Helper T cells bind to the antigen on the antigen presenting cell. This helper T cell stimulates a specific B cell. The B cell then clones or divides by mitosis and forms plasma cells that release antibodies. Question 13. What is an antigen? It is a foreign protein that stimulates an immune response or the production of an antibody. What is an antibody? A protein specific to an antigen, produced by plasma B cells.